Now, today's topic is the how ATP is generated. You know the ATP is the currency of energy. What is currency? Every country has the name of currency. Money is the driving force of the society. Money, আমাদের এখানে কারেন্সি হচ্ছে টাকা, রুপি, ইউএসএতে ডলার, রাশিয়াতে রুবল. In the living organism, the driving force is the ATP. ATP is the driving force in the living organism, in all living organisms, not only the human beings. And ATP is the full name is the adenosine triphosphate. That means it is a nucleotide. You know the nucleotide. Already nucleotide has been taught. It is the composed of nitrogenous base, sugar and phosphate. That is, if it is adenosine is a nucleoside. Nucleoside is the composed of the base and sugar. Here base is the adenine. The sugar is the ribose. Pentose sugar, ribose. And it is connected to triple phosphate. It may be adenosine, phosphate, phosphate. This is the alpha, this is the beta, this is the gamma phosphate. And the italic, not the straight bond, it means it is the high energy compound. High energy bond. This bond is called high energy bond. This is the normal bond. This is ester bond. This is acid anhydride bond. And this is acid anhydride bond. ATP becomes the currency of energy, not the ADP, adenosine diphosphate, nor the AMP, adenosine monophosphate. These are not currency. ATP is the currency. And our primary aim of the metabolism, not only the primary aim, actually it is the ultimate aim of the metabolism, is the generation of the energy in the form of ATP. Primary or ultimate aim, whatever you call, of the metabolism, all sorts of metabolism. It may be carbohydrate, it may be protein, it may be lipids. All the sorts of metabolism, ultimate aim is the generation of the energy in the form of adenosine triphosphate. Now what are the methods of ATP generation? What are the methods of ATP generation? Methods of ATP generation. Of course, 95% of the total ATP used up in our body, 95% of the total ATP, that is major ATP, is formed by oxidative phosphorylation. Major means it is approximately 95% of the total ATP. 95% of the total ATP is generated by oxidative phosphorylation. What is it? It is a process by which ATP is produced as a result of transfer of electrons through electron transport chain. In the abbreviated form is the ETC, transfer of electrons through to ETC. ট্রান্সফারটা কোথা থেকে হয়? From NADH and FADH2. You are already familiar with these molecules, NADH and FADH2. Collectively এটাকে বলে reducing equivalent. These are called reducing equivalents because it contains electrons, a pair of electrons. A process by which ATP is produced as a result of transfer of electrons through ETC. Transfer of electron কোথেকে হয়? From NADH to oxygen, which is the ultimate acceptor of the electrons. Electrons are captured by oxygen, and oxygen itself gets reduced by capturing electron. Gain of electron means reduction, loss of electron means oxidation. And you know, when there is oxidation, there is a concomitant reduction. When somebody gets oxidized, somebody must get reduced. This is the principle of oxidation and reduction. This is the major portion, and another part substrate level phosphorylation. Whether you are substrate level phosphorylation, though it is minor, it is important. কোথায় ইম্পর্টেন্ট? সাবস্ট্রেট লেভেল ফসফোরিলেশন কোথায় ইম্পর্টেন্ট? সবারই তো মাইটোকন্ড্রিয়া থাকে না, ইলেকট্রন ট্রান্সপোর্ট চেইন তো মাইটোকন্ড্রিয়ার ভেতরে আছে। তাহলে মাইটোকন্ড্রিয়া যাদের নেই তারা কি বাঁচবে না? তাদের বাঁচার অধিকার আছে। যাদের মাইটোকন্ড্রিয়া নেই, কোন সেলের মাইটোকন্ড্রিয়া নেই? আরবিসি, ভেরি ইম্পর্টেন্ট সেল। RBC has no mitochondria, but it generates ATP for its survival. How it can get ATP? It can get ATP by through substrate level phosphorylation. That means phosphorylation occurs without help of mitochondria, without oxygen, at the level of the reactants or the level of the substrates. Substrate level phosphorylation means generation of the ATP by phosphorylation of ADP in absence of mitochondria, in absence of oxygen, at the level of substrates. An example take it. There are 3 examples in our body, only 3, not more than 3. What are the 3 examples of substrate level phosphorylation? 2 in the glycolysis, 1 in the Krebs cycle. 2 in the glycolysis কি আছে? কোন স্টেপ? phosphoglycerate kinase. Number 2, pyruvate kinase. এই দুটো এনজাইমেটিক স্টেপ আছে যেখানে এটিপি তৈরি হচ্ছে ফ্রম এডিপি। See the reaction. I will not tell the reaction here. See the reaction where ADP is converted to ATP. And this is in the glycolysis. Substrate level phosphorylation short note আছে in many times, very important. Oxidative phosphorylation long question আসবে, long question আসবে oxidative phosphorylation. আর substrate level phosphorylation হচ্ছে short note. Number 3 হচ্ছে গিয়ে in Krebs cycle 1 step succinate thiokinase, all kinase. এটা হচ্ছে PCA cycle. These three are the examples of the substrate level phosphorylation where ATP is generated from ADP without help of mitochondria, without need of oxygen. And that is important for the cells which are deprived of mitochondria. Now I will come to the original topics. How metabolism is linked to phosphorylation? How metabolism is linked to oxidative phosphorylation? Metabolism means there are some nutrients in the foodstuff which gets metabolized. Nutrients are foodstuffs. What are that? Protein, carbohydrate, lipid. These are the three important nutrients which we intake as a foodstuff. Carbohydrate, protein and lipid, all sorts of metabolism we have finished. Now what happens in protein? From protein and from carbohydrates, what are the simple form? By digestion and absorption. By digestion and absorption, what are the simple form? It is the amino acids. Monomer. Simple form is a monomer. Where carbohydrate is actually we take in the form of starch mainly. And the starch is converted to glucose by the amylase and also all other enzymes necessary for conversion of the starch to glucose, you know. That means glucose. And here lipid is converted to triglyceride, is converted to glycerol plus fatty acids. Now glucose is converted to pyruvate. Pyruvate is converted to acetyl-CoA, and which is the common intermediate. We can get pyruvate from amino acids. Not all amino acids gives rise to pyruvate, but some of the amino acids give rise to pyruvate, particularly the alanine. you can get acetyl CoA, you can get glycerol can be converted to pyruvate. How glycerol can be converted to pyruvate? Do you know? Fatty acid is converted to acetyl CoA by steps of beta oxidation. It is the glycolysis. It is the PTHC. Now, what is the fate of the acetyl CoA? Acetyl CoA enters into the Krebs cycle. That is, acetyl CoA gets oxidized to carbon dioxide. One molecule of acetyl CoA is oxidized to 2 molecules of carbon dioxide through citric acid cycle. This is all of you know. I am just recapitulating for linking oxidative phosphorylation to metabolism. I am just reproducing metabolism. All of you know it and how it is linked to oxidative phosphorylation. This is my concern. Acetyl CoA enters into the TCA cycle. In the mitochondria. Then acetyl CoA gets oxidized to carbon dioxide. So there must be oxidizing agent. There must be some oxidizing agent which will get reduced. Which one is the oxidizing agent of the Krebs cycle? Which one is the oxidizing agents of the Krebs cycle? Which itself gets reduced at the expense of the oxidation of the acetyl CoA? NAD. One is the NAD, another is the FAD. These are oxidizing agents, and oxidizing agents itself gets reduced at the expense of oxidation of someone. Oxidizing agents mean it gets itself reduced and it oxidizes others. It oxidizes acetyl CoA to carbon dioxide, but it gets reduced. It itself, because simultaneously what is oxidation and reduction. That means NAD is the coenzyme derived from the niacin. FAD is the coenzyme derived from riboflavin. Flavin, adenine dinucleotide, nicotinamide, adenine dinucleotide. These are the coenzymes derived from vitamin B, and this gets reduced at the expense of oxidation. That means it will get NADH2, this is gets FADH2. NADH, you remember NADPH is also reducing equivalence. But there are lots of difference between the NADH and NADPH in terms of function. NADH is a reducing equivalence, NADPH is also reducing equivalence. But the purpose of the NADH is to produce energy. Purpose of the NADPH is the biosynthesis of the fatty acid, of cholesterol, glutathione, and many more biomolecules. NADPH and NADH, these are two different things. These are more or less similar in structure, except one additional phosphate is present in NADPH, but NADPH has no role in oxidative phosphorylation or energy generation. Remember, NADPH কখনো লিখবে না in oxidative phosphorylation, and both NADH and FADH2 are collectively called reducing equivalents, from which electrons pass through the respiratory chain. তাহলে নেক্সট উইল কাম, এটা বড় নয়, নেক্সটলি আমাকে আগেরটা মুছতে হবে। লিঙ্ক এখনো কমপ্লিট হয়নি। I have to complete the link. তাহলে TCA সাইকেল থেকে আমরা পাচ্ছি NAD FAD, NADH FAD H2. Now, what are the fates of the NADH2 and FADH2? Fates of the NADH and FADH2 is that it will get reoxidized. It will get reoxidized to NAD and FAD. যদি NAD and FAD যদি সব সময় কন্টিনিউয়াসলি ইউজ হয় to operate Krebs cycle, to operate beta oxidation, যদি NAD and FAD is exhausted, then in the a certain time will come when there will be no Krebs cycle, no beta oxidation. So, but nature has made provision to regenerate NAD from NADH and FAD from FADH2. Nature has made process by which NADH will be converted to NAD and NAD FADH2 will be converted to FAD, and that process is the oxidative phosphorylation. তাহলে এটা TCA সাইকেল, এখান থেকে কার্বন ডাই অক্সাইড বেরোবে, it coming from the acetyl CoA. Now, electron transport chain and oxidative phosphorylation. FADH2 আসবে, ঢুকবে, বেরোবে FAD হিসাবে, NADH2 ঢুকবে, বেরোবে NAD হয়ে, তাহলে এইভাবে NAD আর FAD আবার রিজেনারেট হলো। তাহলে পারপাস অফ দ্য অক্সিডেটিভ ফসফোরিলেশন আর দ্য টু. Major function is the ATP generation, and one of the another important function is the regeneration of the NAD and FAD. Major function of the oxidative phosphorylation is the ATP generation. But in case of NAD and FAD, I call it regeneration, because NAD and FAD is already present beforehand, but it is exhausted. It becomes regenerated. Regeneration of the reducing coenzyme, regeneration of the coenzyme NAD and FAD, and the generation of the ATP. These are the two most important functions of the respiratory chain and oxidative phosphorylation, which occurs within the mitochondria, and so mitochondria is known as powerhouse of the cell. Why mitochondria is called powerhouse of the cell? Because without mitochondria we cannot get energy. Without mitochondria we cannot get energy. Only a portion of the energy we can get substrate level phosphorylation, but it is not sufficient to maintain life. Now the question is, when NADH2 and NADH and FADH2 gets oxidized to NAD and FAD, which one gets reduced? Which one gets reduced? যখন NADH এবং FADH2 reoxidation হবে, তখন somebody gets reduced. Which one? Which one? It is oxygen. Oxygen, reduction of the oxygen to water at the expense of oxidation of the NADH and FADH2. Oxidation of the NADH and FADH2 occurs at the expense of reduction of oxygen, molecular oxygen to water. Now, concomitantly, by transfer of electrons, by the definition of the oxidative phosphorylation, we have told it is a process of ATP generation How from ATP will be generated? ATP will be generated from ADP by phosphorylation. Phosphorylation of the ADP and oxidation of the foodstuffs. So, term oxidative phosphorylation. Oxidation of the foodstuffs and phosphorylation of the ADP is the result of the ATP generation. When electron passes through the electron transport chain, I am coming on that, which is the major topic or main topic of discussion today. এইখানে ADP gets phosphorylated, তাহলে একটা inorganic phosphate লাগবে, ATP will be produced. And ATP will be produced for consumption, for utilization. ATP যখন প্রডিউস হয়েছে তখন এটা ইউটিলাইজ হবে। Unless it is utilized, it need not be produced. How ATP is utilized? How ATP is utilized and what are the reactions of ATP utilization? Reaction of the ATP utilization, it is the hydrolysis of the ATP to form ADP. ADP regenerate করবে, ATP again it will phosphorylate to form ATP. ATP will use that to form ADP and that is known as ATP-ADP cycle. What is ATP-ADP cycle? ATP তৈরি হলো, ATP utilize হলো. This is ATP formation step. And this is ATP utilization step. ATP formation and ATP utilization occurs constantly. This is the hydrolysis. ATP formation, ATP utilization occurs simultaneously. ATP তৈরি হচ্ছে, আমরা foodstuffs খাচ্ছি, oxidative phosphorylation হচ্ছে, ATP তৈরি হচ্ছে, ATP used up হচ্ছে. What are the methods of ATP used up? ATP কোথায় কাজে লাগে? ATP কাজে লাগে তিনটে মেজর জায়গায়, যেখানে ATP কাজে লাগে। 1/3, 1/3, 1/3, 1/3, 1/3, 1/3. ATP utilization, and this is day-to-day activity. Exercise, walking, talking, digestion, exercise, respiration, in all forms of the work in life. All forms of the work in life requires ATP. All forms of work in life requires ATP, and which is utilized and which is the first category of the utilization. Second category of the utilization, 1/3 of the ATP will be reductive biosynthesis. Term is very important. Reductive biosynthesis. Reductive biosynthesis means biosynthesis of the fatty acid, biosynthesis of the cholesterol, biosynthesis of the glucose, biosynthesis of the protein, synthesis of the DNA. All synthesis requires ATP. All synthesis of the biomolecules, whatever may be the nature of the biomolecules. It may be glucose, it may be protein, it may be DNA, it may be fatty acid, it may be cholesterol. Whatever may be the biomolecules, for the formation of which requires ATP and which is the 1/3 ATP consumption. টোটাল এটিপি কনজাম্পশনের 1/3 চলে গেল ডে টু ডে অ্যাক্টিভিটির জন্য। হাঁটছি, এক্সারসাইজ করছি, খাচ্ছি, ঘুমাচ্ছি, নিঃশ্বাস নিচ্ছি, ক্যালপিটেশন হচ্ছে, হার্টবিট হচ্ছে, ফর দ্যাট 1/3 and 1/3 biosynthesis. And third category is very important to maintain electrolyte balance, particularly sodium and potassium. For maintenance of the sodium and potassium equilibrium, we require a pump, that is sodium potassium ATPase, and this Operation of this pump, sodium potassium ATP requires 1/3 of the ATP, total ATP. তাহলে 1/3 will be to operate sodium, potassium, ATP, it is a pump. Pump means it requires some energy to operate. All pumps require some energy to operate. Otherwise, it will not operate. So, 1/3 is day-to-day activity, 1/3 is reactive biosynthesis, 1/3 to operate the pump. This is the methods of utilization of the ATP. ATP is continuously produced by oxidative phosphorylation, and ATP is used up by these three processes. And this is the sequence of the metabolism of the foodstuffs to ultimately generation of the ATP. All the steps starting from the metabolism of the foodstuffs to generation of the ATP are collectively called biologic oxidation. Biologic oxidation is a terminology which starts from oxidation of the foodstuffs and which ends in generation of the ATP. It is the oxidation of the foodstuff থেকে আরম্ভ করে সমস্ত স্টেপ অক্সিডেশন অফ দ্য ফুডস্টাফের জন্য যা যা সমস্ত স্টেপ লাগে ক্রেবস সাইকেল গ্লাইকোলাইসিস অ্যামিনো অ্যাসিড অক্সিডেজ বিটা অক্সিডেশন অল দ্য metabolic processes along with oxidative phosphorylation and ATP generation are altogether called biologic oxidation, and major part of the biologic oxidation is contributed by oxidative phosphorylation. Oxidative phosphorylation is biologic oxidation, the overall oxidation in our biological system, overall oxidative process in our biological system, and oxidative phosphorylation is the major portion of the biological oxidation which generates ATP. Biologic oxidation and oxidative phosphorylation, there is a difference. Oxidative phosphorylation is a major part of the and predominant part of the biologic oxidation. Now I am coming to the electron transport chain. Electron transport chain means electrons which is present in the reducing equivalence, that is NADH and another is FADH2. There are contains a pair of electrons. Reducing equivalence contains pair of electrons. কি আছে reducing equivalence? NADH, reducing equivalence contain a pair of electrons, and this pair of electrons are transferred. Pair of electrons are transferred. Electron কখনো এক জায়গায় বসে থাকবে না. Electron will transfer. These electrons are transferred from NADH FADH2 to oxygen, which is the final acceptor of the electron. Oxygen is the final acceptor of the electron. আর ইনিশিয়াল ডোনার অফ দ্য ইলেকট্রন হচ্ছে গিয়ে NADH and FADH2. এই ইনিশিয়াল ডোনার অফ দ্য ইলেকট্রন অ্যান্ড ফাইনাল অ্যাকসেপ্টার অফ দ্য ইলেকট্রন এর মাঝে অনেকগুলো কম্পোনেন্ট আছে। ইনিশিয়াল ডোনার অফ দ্য ইলেকট্রন and final acceptor of the electron, that is oxygen. In between these there is a component which is known as electron transport chain. Electrons are transferred from NADH or FADH2 to oxygen through properly arranged. This is very important. Not haphazardly arranged, properly arranged set of collectively called electron transport chain, or in abbreviation we write ETC. এটাকে আরো রেসপিরেটরি চেনও বলে। সিমিলার থিং, ইলেকট্রন ট্রান্সপোর্ট চেন এন্ড রেসপিরেটরি চেন or similar. Situated in কোথায় থাকছে? Inner membrane of the mitochondria. Inner membrane of mitochondria. I am abbreviated form mitochondria MC. ETC electron transport chain. তাহলে ইলেকট্রন ট্রান্সপোর্ট চেন কাকে বলবো? ইলেকট্রনস আর ট্রান্সফার্ড কোত্থেকে আসছে? ফ্রম রিডিউসিং ইকুইভ্যালেন্স ফ্রম NADH and FADH2 কোথায় যাচ্ছে? আর ফাইনালি টু অক্সিজেন। কিভাবে যাচ্ছে? একটা সেট অফ ক্যারিয়ার্স, সেট অফ অ্যাকচুয়ালি কম্পাউন্ডস না বলে ইট শুড বি কল ইলেকট্রন ক্যারিয়ার্স, set of electron carriers, more appropriate, set of electron carriers, a properly arranged set of electron carriers যেটা আছে in between NADH and oxygen or in between FADH2 and oxygen, এই সেটটাকে বলছে রেসপিরেটরি চেন or electron transport chain. তাহলে ইলেকট্রন ট্রান্সপোর্ট চেন কোথায় থাকছে? ইন দ্য ইনসাইড অফ দ্য মাইটোকন্ড্রিয়া। ইনসাইড অফ দ্য মাইটোকন্ড্রিয়া অনেকগুলো ঘটনা ঘটে। What are the events occurring in the mitochondria? You know the mitochondria is the powerhouse of the cell. I will just recollect the events in the mitochondria. What are the events? What are the structure of the mitochondria? Where electron transport chain is situated? মাইটোকন্ড্রিয়া দেখতে কিরকম? এটা ওভাল শেপের, একটা ওভাল শেপের স্ট্রাকচার। আই এম টকিং অ্যাবাউট মাইটোকন্ড্রিয়াল স্ট্রাকচার। একটা মাইটোকন্ড্রিয়া হলে কিন্তু মাইটোকন্ড্রিয়ন বলে, সিঙ্গেল ফর্ম হচ্ছে মাইটোকন্ড্রিয়ন। when mitochondria it is a plural form, remember মাইটোকন্ড্রিয়া আমরা ইউজুয়ালি বলি, কিন্তু it is not single mitochondria, it is a many more. এটার দুটো ফোল্ড আছে, একটা ইনার মেমব্রেন, একটা আউটার মেমব্রেন। ইনার মেমব্রেন is extremely folded, extremely folded inner membrane. And the space encircled by the inner membrane. তাহলে এটা হচ্ছে কি আউটার মেমব্রেন, আউটার মেমব্রেন, এটা হচ্ছে কি ইনার মেমব্রেন, এটা হচ্ছে গিয়ে ম্যাট্রিক্স। তিনটে পার্ট, আর মাঝখানে যে স্পেস আছে ইন বিটুইন দ্য টু, এটাকে বলছে ইন্টার মেমব্রেনাস স্পেস। দিস ইজ দ্য স্ট্রাকচার অফ দ্য মাইটোকন্ড্রিয়া। তুমি রেসপিরেটরি চেইন তাহলে কোথায় থাকছে? আমি একটু বড় করে লিখছি, আঁকছি এটা। Inner membrane is extensively folded. Why? Why it is extensively folded? To increase the surface area. Why surface area is required to be increased? To accommodate all the components of the electron transport chain in massive amount. Electron microscope chain, inner membrane is extensively folded to increase the surface area, to accommodate enormous number of ইলেকট্রন ট্রান্সফার চেন। ইলেকট্রন ট্রান্সফার এটা কিভাবে থাকে? দিস ইজ ইলেকট্রন ট্রান্সফার চেন। দিস স্টার্টেড পোর্শন ইজ দ্য ইলেকট্রন ট্রান্সফার চেন। These are the inner membrane which is started with electron transport chain. Sufficient number of electron transport chain is required to accommodate, so inner membrane is exfolded. It is the matrix. What are the events in the matrix? Events in the matrix কি হয় বলো? PCA cycle. PCA cycle এনজাইম থাকছে। কি আর কি এনজাইম থাকে? বিটা অক্সিডেশন। আর কি থাকে? পিডিএইচ কমপ্লেক্স। আরো থাকে অ্যামিনো অ্যাসিড অক্সিডেজ, আরো অনেক থাকে। দিস আর দ্য থ্রি মেন যেখান থেকে আমরা রিডিউসিং ইকুইভ্যালেন্স পাই। তিনটে মেন জায়গা, একটা হচ্ছে ক্রেবস সাইকেল, একটা হচ্ছে বিটা অক্সিডেশন, একটা হচ্ছে পিডিএইচসি। এখান থেকে আমরা রিডিউসিং ইকুইভ্যালেন্স পাই আইদার ইন দ্য ফর্ম অফ NADH or in the form of FADH2. And these are the respiratory chain or the electron transport chain. Intermembrane space. Intermembrane space এ থাকে দুটো এনজাইম থাকে, ক্রিয়েটিন কাইনেজ একটা অ্যাডেনাইলিন কাইনেজ। এগুলো থাকে ইন্টারমেমব্রেন স্পেসে। আর আউটার মেমব্রেনে থাকে ফর অ্যাক্টিভেশন অফ দ্য ফ্যাটি অ্যাসিড যে এনজাইম। আমি সবগুলোকে একসঙ্গে বলছি, আমি সব ইভেন্টস। ফ্যাটি অ্যাসিড অক্সিডেশনটা কোথায় হয়? ফ্যাটি অ্যাসিড অক্সিডেশন অকার্স ইন দ্য আউটার মেমব্রেন বাই দ্য এনজাইম অ্যাসাইল কোয়ে সিনথেটেজ। তাহলে অ্যাসাইল কোয়ে সিনথেটেজ কোথায় দেখছে? আউটার মেমব্রেনে। কিন্তু বিটা অক্সিডেশনের এনজাইমস গুলো কোথায় থাকবে? ম্যাট্রিক্সে। ক্রস করতে হবে, তার জন্য দরকার কার্নিটিন। আমি ওইদিকে যাচ্ছি না। I am now focusing to our main topics, that is oxidative phosphorylation and electron transport chain. ইলেকট্রন ট্রান্সপোর্ট চেইন ডেফিনেশন বলেছি, ইলেকট্রন ট্রান্সপোর্ট সিচুয়েশন বলেছি, এবং মাইটোকন্ড্রিয়াকে কেন পাওয়ার হাউস অফ দ্য সেল সেটাও আমি বলেছি। পাওয়ার হাউস অফ দ্য সেল, why mitochondria is called power house of the cell? You must explain it, because it is the major site of the ATP generation. প্রত্যেক ATP generation হয়, you have to cite that examples. You have to draw the structure and you have to mention the sites, outer membrane, inner membrane and in mitochondrial matrix. এই মাইটোকন্ড্রিয়াল ম্যাট্রিক্সে যে ক্রেব সাইকেল হয়, এই ক্রেব সাইকেল থেকে NADH FADH2 তৈরি হয়। এই জন্য মাইটোকন্ড্রিয়াল ম্যাট্রিক্স আর ইনার মেমব্রেন একদম কাছাকাছি আছে, যাতে মাইটোকন্ড্রিয়াল ম্যাট্রিক্সে যে রিডিউসিং ইকুইভ্যালেন্স তৈরি হয় তারা যাতে খুব চট করে ইলেকট্রন ট্রান্সপোর্ট চেনে চলে যেতে পারে, তার জন্য মাইটোকন্ড্রিয়াল ম্যাট্রিক্স আর ইলেকট্রন ট্রান্সপোর্ট চেন আর ফেসিং দ্য মাইটোকন্ড্রিয়া, ফেসিং দ্য matrix of the mitochondria. This is why the close proximity of the matrix and electron transport chain. Now I will draw the diagram first. ইলেকট্রন ট্রান্সপোর্ট চেন আমি আগে ডায়াগ্রামটা আঁকি, তারপরে তোমাকে আমি ডেসক্রাইব করছি। পরে বলছি কোনটা কি। এটাকে বলছে কোএনজাইম কিউ or ubiquinone। এই যে সাইটোক্রোম, দিস ইজ সাইটোক্রোম, CIT means cytochrome, ATS means iron sulfur protein. There are many cytochromes. I am telling that on I am describing succinate dehydrogenase, which is the complex 2. এখানে থাকবে FAD ATS, this is complex 1, this is complex 2, this is complex 3, this is complex 4. One, two, three will be in a Roman letter. 1, 2, 3 will be in Roman. তাহলে starting material কি বলেছিলাম? Starting material is the NADH or FADH2. NADH comes from here and FADH2 comes from complex 2. That is NADH is converted to NADH is deoxidized to NAD and oxygen is converted to water. In between this one and this one, all the components which are properly arranged, not haphazardly. Complex 2টা complex 1 এর জায়গায় থাকলে হবে না, কিন্তু complex 4টা complex 1 এর জায়গায় থাকলে হবে না। It is properly arranged set of electron carriers. Properly arranged set of electron carriers. That is electron transport chain. এইটা হচ্ছে কি electron transport chain. And these are the all components. Maximum number of components of the electron transport chain are organized into complexes, except two. One is coenzyme Q, another is cytochrome C. এই দুটো ছাড়া যত components আছে, they are organized either in complex 1 or in complex 2 or in complex 3 or complex 4. Remember, these two are not within complex. One is Q and one is the C. These are not within the complex, it is outside the complex. And what is the nature of the complex? All the complexes are integral membrane protein. All the complexes are integral membrane protein of the inner mitochondrial membrane. Remember, all the complexes 1, 2, 3, 4 are integral transmembrane protein of the inner mitochondrial membrane. But cytochrome C, which is also a protein, it is peripheral protein. It is mobile. All the complexes are embedded, integral proteins are embedded in the membrane. All the complexes are integral membrane proteins are embedded in the inner membrane and are fixed. But cytochrome C is a protein but not fixed. It is peripheral protein. Q is not at all protein. Q is a lipid in nature. তার সমস্ত কমপ্লেক্সের যে ইলেকট্রন ট্রান্সপোর্ট চেনের যতগুলো মেম্বার আছে তার মধ্যে একটা লিপিড মেম্বার আছে। দ্যাট লিপিড মেম্বার ইজ কোএনজাইম কিউ অর ইউবিকুইনন। সমস্ত কম্পোনেন্টস কিন্তু প্রোটিন ইন নেচার এক্সেপ্ট দ্য কিউ। সমস্ত কমপ্লেক্স কিন্তু ইন দ্য সিচুয়েটেড ইন দ্য ইনার মাইটোকন্ড্রিয়াল মেমব্রেন except the cytochrome C, which is a peripheral protein. Complex 1 কে আমরা কি বলি? Complex 1 কে আমরা বলি NADH থেকে Q এ যাচ্ছে, সেই জন্য NADH dehydrogenase বলি, আবার NADH-Q। oxidative, এটা কোএনজাইম Q আর ইউবিকুইনন। এইটাকে বলবো কিউ cytochrome C, oxidative, this is cytochrome oxidase। এই যে এখানে সমস্ত কিন্তু সাইটোক্রোম বলেছি, দিস আর অল কল সাইটোক্রোম। এখানে একটা সাইটোক্রোম আছে, এখানে একটা সাইটোক্রোম আছে, এখানে একটা সাইটোক্রোম আছে, এইখানে সাইটোক্রোম বি আছে, এখানে সাইটোক্রোম C1 আছে। দিস ইজ সাইটোক্রোম C, দিস ইজ সাইটোক্রোম A and this is cytochrome H. এই সমস্ত what are the cytochromes? What are the cytochromes? Cytochrome is a protein in nature and actually it contains a prosthetic group. Which is the prosthetic group of the cytochromes? Do you know the cytochromes? Which prosthetic group is this content? It is a heme, iron, heme. Not only iron is present in iron sulfur protein, but it is not the heme. Iron is present in iron sulfur protein, which is called non-heme iron. But iron present in the hemoglobin, iron present in the cytochrome C, iron present in the catalase, iron present in peroxidase, that is heme iron. তাহলে heme containing protein, this is asked in the examination. What are the heme containing proteins? Heme containing proteins, you must tell first hemoglobin, myoglobin, and then if you stop, what are the other proteins? What are the other proteins? You tell them cytochromes, you tell the enzymes, cytochrome oxidase, you tell the enzymes, catalase, you tell the enzyme, peroxidase. These are the all biomolecules containing heme as a content. These are the heme containing proteins, which is known as, collectively known as hemoproteins. Hemoproteins are heme containing proteins. Hemoglobin, myoglobin, cytochromes, catalase, peroxidase. Remember, I am not writing. Heme containing proteins are frequently asked, and you have to mention hemoglobin, myoglobin, catalase, peroxidase, and cytochromes. These are properly arranged. কেন কিভাবে অ্যারেঞ্জ করা হয়েছে? কিভাবে অ্যারেঞ্জ করা হয়েছে? According to the electron affinities. Electron affinities কার বেশি? সবচেয়ে বেশি ইলেকট্রন, highest electron affinity is the oxygen, which is the ultimate electron acceptor. Ultimate electron acceptor oxygen has the highest electron affinity. এবং electron affinity of the left side, that is NADH, is the lowest. তাহলে in ascending order of the electron affinities এগুলো অ্যারেঞ্জ করা হয়েছে. In ascending order of the electron affinities, electron transport chain are arranged. তাহলে ascending order of the electron affinity means a terminology we use, that is redox potential. Redox potential is the measurement of the electron affinity. তাহলে increasing redox potential, increasing electron affinities, redox potential যার বেশি তার ইলেকট্রন অ্যাফিনিটি সবচেয়ে বেশি। তাহলে এইভাবে অ্যাসেন্ডিং ম্যানার অফ দ্য ইনক্রিজিং রিডক্স পোটেনশিয়াল এটাকে অ্যারেঞ্জ করা হয়েছে, যার জন্য ইলেকট্রন ট্রান্সফার হয়। ইলেকট্রন ট্রান্সফার হচ্ছে মানে this one gives electron to the complex one, and complex one itself gets reduced after accepting electrons. কিন্তু রিডিউস অবস্থায় থাকবে না, সে থাকবে না, কারণ ও আবার সেই ইলেকট্রনটা ট্রান্সফার করে দেবে। কাকে করবে? কিউকে। তাহলে কমপ্লেক্স ওয়ানের কাজটা কি? ইট অ্যাকসেপ্টস ইলেকট্রন ফ্রম NADH and passes to the Q. কমপ্লেক্স টুর কি কাজ? ইট অ্যাকসেপ্টস ইলেকট্রন ফ্রম FADH2। এখানে FADH2 তৈরি হবে and it passes the electrons to the Q. FADH2 থেকে Q আসবে, NADH কেও Q আসবে, সেই জন্য ইট ইজ কল্ড NADH Q অক্সিডোরিডাক্টেজ। এটাকে বলে সাকসিনেট Q অক্সিডোরিডাক্টেজ। সাকসিনেট থেকে Q এ যাচ্ছে, সেই জন্য সাকসিনেট Q ডিডাক্টেজ আর অক্সিডোরিডাক্টেজ, whatever you call. Complex one transfers electron from NADH to Q. Q gets reduced, but it cannot get reduced in all time. It will get oxidized by passing its electron to this complex. Loss of electrons means oxidation, gain of electron means reduction. তাহলে একই সঙ্গে সাইমালটেনিয়াসলি অক্সিডেশন এবং রিডাকশন ঘটছে, এবং সাইমালটেনিয়াসলি অক্সিডেশন রিডাকশন ঘটতে যাচ্ছে বলে ইলেকট্রনস আর ট্রান্সফার from NADH to final acceptor oxygen. আবার ইলেকট্রন নিল, ইলেকট্রন নেওয়ার পর এখান থেকে ইলেকট্রন নিয়ে রিডিউস হলো, রিডিউস হওয়ার পরে কি করবে? এই ইলেকট্রনটাকে আবার সাইটোক্রোম সিতে ট্রান্সফার করলো। যখন সাইটোক্রোম সিতে ট্রান্সফার করলো, এটা রিডিউস হয়ে গেল, এটা অক্সিডাইজ হয়ে গেল। আবার সাইটোক্রোম সি কি করবে? সেই যে অক্সিজেন ইলেকট্রনটা পেয়েছে ওর কাছ থেকে, আগের মেম্বারের কাছ থেকে, সে নেক্সট মেম্বারকে ইলেকট্রনটা ট্রান্সফার করবে এবং নিজে আবার অক্সিডাইজ হয়ে যাবে, একে রিডিউস করবে। রিমেম্বার, এই অক্সিডেশন রিডাকশন মানে তুমি বেসিক প্রিন্সিপল অফ দ্য অক্সিডেশন মিন্স লস অফ ইলেকট্রন মিন্স অক্সিডেশন, গেইন অফ ইলেকট্রনস আর দ্য অক্সিডেশন। যে ইলেকট্রন অ্যাকসেপ্ট করবে সে রিডিউস হচ্ছে, যে ইলেকট্রন ডোনেট করছে সে রিডিউস হচ্ছে, সে অক্সিডাইজ হচ্ছে। যে ইলেকট্রন ডোনেট করছে সে অক্সিডাইজ হচ্ছে, যে ইলেকট্রন অ্যাকসেপ্ট করছে সে রিডিউস হচ্ছে। এবং অক্সিডেশন এবং রিডাকশন সবসময় ঘটতেই থাকছে বিকজ অফ দিস simultaneous oxidation and reduction of all the components. Electrons are transferred from NADH to oxygen. আচ্ছা, এরপরে ক্লাস আছে তোমাদের, ছেড়ে দিই তাহলে নাকি? সোমবার আমি নেবো। আমি Any query please clarify. কোথাও কোনো কোয়েরি আছে? এ পর্যন্ত আমি যা পড়ালাম, আমি পড়িয়েছি বায়োলজি অক্সিডেশন, আমি পড়েছি মেটাবলিজম অফ ফুডস্টাফ লিঙ্ক টু অক্সিডেটিভ ফসফোরাইলেশন, আমি পড়েছি কম্পোনেন্টস অফ দ্য ইলেকট্রন ট্রান্সফার চেইন, আমি পড়িয়েছি রেডক্স পোটেনশিয়াল, আমি পড়িয়েছি রেডক্স পেয়ার। আই উইল কাম ডিটেলস ইন্টেলিজেন্স ডে হোয়াট ইজ রেডক্স পোটেনশিয়াল এন্ড রেডক্স পেয়ার, এই কোথায় এবং কেন ওটা প্রপারলি সেট আছে আমি বলেছি। কোনটা ইন্টিগ্রাল প্রোটিন, কোনটা পেরিফেরাল প্রোটিন, কোনটা লিপিড, কোনটা প্রোটিন, কোনটা কোথায় থাকে, আই হ্যাভ এক্সপ্লেন। ইফ এনি কোয়ারি টেল মি।
Today we perform quantitative estimation of serum creatinine. Okay. But there is slightly change in the calculated formula, that is OD of test upon OD of standard minus OD of blank upon OD of blank upon concentration of standard upon volume of sample multiplied by 1000. Don't it turned off? I'm not joking here. Yes, this is Japping method. Quantitative estimation of Japping method. Previously I will take the class. What I write you the calculated formula OD of test upon OD of standard multiplied by concentration of standard. That is 2. But there is slightly change. OD of test upon OD of standard minus OD of length. Then directly multiply by 2. How we get 2? From this calculated formula. Concentration of standard upon volume of sample multiplied by 1000.
सर सेकेंड फॉर्मूला में ओडी ऑफ ब्लैंक बाय ओडी ऑफ ब्लैंक दोनों तो कट जाएगा ना।
कट जाने तो वो फॉर्मूला है, लेकिन काट दो तो आपको जो करना है वो करो। बट दिस इज द फॉर्मूला।
सर असली फॉर्मूला क्या है?
Without discussing anything, just see the carefully the formula. This is OD of test minus OD of blank upon OD of standard minus OD of blank multiply by 2.
सर वो उस टाइम इतना गैप था ना, हमको लगा कि मतलब माइनस ओडी ऑफ ब्लैंक
इसलिए तो कह रहे हैं दिमाग खोल के रखो, फालतू मोबाइल में मत लगाओ, केक और वो सब करना। So finally here what we calculate concentration of creatinine is equals to OD of test upon OD of standard by 2. ठीक है, because students see we perform here this test on protein free filtrate PFF. Protein free filtrate. This is the sample which we obtained from the blood, that all the proteins we filtered out, and the solution which comes we call it that protein free filtrate. Okay. For this we make the concentration of multiply by 2. Okay. Now all of you here are appearing, take your notebook and go to lab. Perform this practice.
What are the points of this? A is basis, upward, medially and backwards. This lesser tubercle faces anterior and the most prominent part in the lower end, that is the projection medial electron line, it faces medially. So these are the points that will determine the anatomical position. Okay. Then one by one we will discuss about the head. Okay. So what are the things you could say? About the head, it will articulate to xenoid cavity of the scapula. So what is the peculiarity of this joint? The cavity, it is disproportional part. Okay, disproportional part. Xenoid cavity is more and small, whereas this one, that is the head of the humerus, is most larger. Okay, so this is disproportional. So, what are the other things you could say about the shoulder joint? It is stable or not? It is most unstable joint of why? It is a variety of ball and socket. It is a variety of ball and socket joint. Okay. So, rather than that, you could see this whole around this head. There is articulation of articular capsule. articular capsule of the shoulder joint. Okay. So one thing is that in the clinical anatomy is that in cases of infants, what is occurring? This capsule of the shoulder joint is more strong than the epiphyseal cartilage. So if there is a fracture of the head of the humerus, what will happen? The head will be separated. Okay, as because this capsular ligament is so much strong, so that it will be separated. This thing you have to remember. Okay. So next point is head is oval, right? What is this present over here? This surface? Glenoid labrum. Glenoid labrum. And this one is for the capsule of the shoulder joint. Okay. Then next one is neck. So first one is the anatomical neck. So what do you mean by anatomical neck? This is the thing. Articular capsule where it is attached. That is also the anatomical neck. Where it is attached. Okay. So that is anatomical neck. Then next one is. So what is the significance of anatomical neck? Is it a complete capsule or incomplete capsule? Why? Incomplete capsule. Why? Which head? Long head of myself brachia is coming over here. So it is a snake neck. Okay, it is incomplete one. Okay. Then next one is the morphological neck. Morphological neck, how to say? So before discussing the morphological neck, you have to elaborate about surgical neck. So what do you mean by surgical neck? So it is the not fracture থেকে হবে না, it is the upper end and the shaft. Midpoint between upper end and shaft. So upper end, this structure is here. This is the greater tubercle. So meeting point of greater tubercle and the shaft. So this is the surgical neck. Tell me which structure we could found over here. Axillary nerve. It will supply. Which muscle it will supply axillary nerve? একটা তো সবাই বলে দেবে আরেকটা teres minor, deltoid and teres minor. Okay, these are most common. Teres minor is supplied by axillary nerve. Any other artery? Posterior. This is posterior surface, right? Posterior circumflex humeral artery. So could you remember about the femur? What are the two arteries? Femur এ এরকমই artery থাকে, circumflex artery. That means it is inserted in the bone. That is medial and lateral circumflex humeral artery, which is the branch of femoral and obturator artery. So here two structures, remember, one is the axillary nerve, another one is the posterior circumflex humeral artery. Okay, then how to see the morphological neck? You have to go just 5 cm above. 5 cm above the surgical neck. Here is the separation between the epiphysis and the diaphysis. So it is not seen, this portion in between structure. Okay. So which humerus we could see? It is not visible, right? In this humerus. Morphological. In which humerus it is prominent? Below 20 years. Okay, young humerus. It is below 20 years because this morphological neck is prominent. Okay. Then next one is about the neck is over, right? So upper end still we are leaving two things. One is greater tubercle, another one is lesser tubercle. Yeah. So at first we will discuss about the greater tubercle. So what are the structure you will see here? There are three muscles. Number one is supraspinatus, another one is supraspinatus. What we do is minus. বাকিটা বের করতে তাই তো বাকিটা পড়ে নেন, ঠিক আছে why it is a part of the rotator cuff. Okay, rotator cuff or tendinous particle you will say in the shoulder joint. What are the four muscles? Okay, another interesting thing is that shoulder joint is the most unstable joint, you know, and the dislocation is very common. But where? Which side? Both side is dislocation machine.
Right side.
Inferior and okay, two side anteriorly and inferiorly. Example হয় cricket এর bowler. Shoulder come out. Okay, so that thing is to occur. So next one is this one is the lesser tubercle. So lesser tubercle, which muscle is attached? Substrate. So what is this in between? So it is also known as bicycle. So it has got two legs, right? One is lateral leg and medial leg. So which structure attached to the which one is lateral? The lateral. Among these, which one is the lateral? এটা হচ্ছে medial. Medial কি থাকবে lateral কি medial কি থাকবে আবার So don't confuse team. Always remember team. That is there is minor in the medial. Okay, there is minor in the medial. Whatever the next one pectoralis major would be laterally. And in between the floor, which one is so what are the other structures that are passing through this bicipital group? Internal license brachii and one artery, another artery. তাহলে এখানে posterior এখানে anterior femoral anterior circumflex femoral. So one of the person told about the transverse femoral ligament. So what is the importance? Where it is present? এরকম একটা জায়গায় আছে, right? Transverse femoral ligament. Because it is fixing the tendon of biceps brachii. Why? Because during extreme abduction, okay, extreme abduction by the latissimus dorsi and pectoralis major would go up, okay, so that this tendon would be fixed. So that is by the transverse femoral ligament. Okay. Then next one is that is about the shaft. So in the shaft, what are the borders? There are three borders and three circles. So how to identify the border? So you have to place only lower end. Okay. Between the capitula and the trochlea, there is a one border is going up. So that is the anterior border. Okay. Then towards the medial epicondyle, another border is going medial border, and towards the lateral epicondyle, that is the lateral border. So what are the surface we are getting? Anteromedial, anterolateral, and posterior surface. Okay, these are the three things. So anterior border is related to which structure? Anterior border is related to which structure? Intravascular septa. is attached to which border? Lateral and medial border. Okay. So anterior border is divided into two surfaces, anterior medial and lateral. So which structure is related? He is right. It is brachialis. Okay, it is brachialis muscle. You understood what are the deep muscle of the arm? Okay, that is the brachialis immediately above, and above that there will be superficial structure biceps. Okay, then medial border here it is also related to. This is the medial border. It is related to thoracobrachialis. Insertion of thoracobrachialis. Okay, now we will come about the lateral border. So if you just trace the lateral border, there you will find one V-shape. It is also known as deltoid tuberosity. Insertion for the deltoid. Now tell me which bursa is present below deltoid? Between the bone and the muscle? Subacromial bursa. So it is subacromial bursa because there will be continuous friction because of deltoid. So এখানে কি bursa থাকে? Subacromial bursa. Okay. Then next one is on the posterior surface. Posterior surface here anterior and posterior. How many heads it has? Lateral, long and medial. Long with kothai lage? Ventral iliac tubercle. Then above here on the posterior surface there is a lateral head, and throughout this whole medial tuber is the medial. Okay, so triceps. Any other muscle on the posterior surface? Yes, anterior. তাহলে এখানে কি লাগবে পোস্টেরিয়র দুটো মাসল একটা লাগবে প্রিভিউ রেডিয়ালিস আরেকটা এক্সটেনসর কার্বোহাইড্রেট laterally কি থাকবে তাহলে brachioradialis above নিচে থাকবে extensor carbohydrate. Okay, so now next thing is which one is the radial group? Could you identify which one is the radial group? Posterior surface. Which one is the radial group? So you have to trace the deltoid tuberosity. Okay, deltoid tuberosity from that input form of one muscle. It is also known as radial group. So what are the content of radial group? Anything else? Which are the structure? Okay, so what is happening? See, suppose this is a radial groove. The radial nerve is running between the groove. Okay, then it is going where? To this groove. Okay, which is present to the lateral supracondylal region. Two structures are present. One is radial nerve, another one is fundabrachial artery. So that the radial nerve is coming from back to front. Okay, and it will start like this structure. Radial root is এটা কি হচ্ছে দুটো দেখতে পাচ্ছিস একটা সুপ্রাকন্ডাইল আর two structure would be present. One is radial nerve, another one is ulnar nerve. Which structure related to medial epicondyle? Ulnar nerve. So it would be which one is the medial epicondyle? So this is the radial nerve. Okay. So now we start with the lower end. Okay. Lower end, what are the structure? First one is the capitula. This one is the trochlea. Okay. So how it is present the radial head of the radius, right? So this is the head of the radius. So what is the anatomical position? Which side radius it is? So how to determine? So radial tuberosity would be always medially and the distal tubercle downwards that is also facing medial. So these are the two things by that way you could identify. Okay. So this is which side radius? Silent process that's right. Right? Radial tuberosity medially happens. So head of the radius would be articulating with the capitula. Okay. So during next level what is happening? It is going to the radial process. Okay. Then what about the ulnar? Which one is the head of the ulnar? So it is doing Shirshasana. Okay. Head is downward, don't confuse. And this is the lower end. Okay. So in lower end, what are the process you need? Which one is here? This C-shape over here. C-shape of next from here. So this is the which one is the organoid process? This one is the organoid process. So this is the rest you will see only next one which is the okay. So see these are the things. So this is the capitula. This one is the trochlear notch that is articulated to the trochlea, and how in the extended elbow this orbital process is going to the orbital process. Okay. So this is the which type of joint? Heme joint. Okay. Now next point is about the ossification. Okay. Ossification, it has got one primary center that is present on the shaft and second secondary. So it has got what are ossification center আছে একটা প্রাইমারি চারটা সেকেন্ডারি রাইট প্রাইমারি সেন্টার কোথায় আছে স্যাটে আছে কখন অ্যাপিয়ার করে এই রিকক রেক্টাইল না ওকে and there is seven secondary center, three in above, right? One for head, one for middle tubercle, one for distal tubercle, and four for lower epicondyle and capitula. And when does it fuse? At the age of 20. Okay. Now write it down clinical aspect. So first thing is the most commonest fracture of humerus is most commonest answer. Most commonest Most commonest fracture of humerus is supracondylar fracture of humerus. Supracondylar fracture of humerus. So that is here. Over here, fracture will be occurring over here. Okay, supracondylar fracture. Then next one is the surgical neck. So for whom this is occurring? Commonest fracture is supracondylar fracture. Is for the newborn and infant. Okay, newborn and infant. Then next fracture is fracture of the surgical neck of humerus. Okay. So when it will occur? If somebody will fall from the outstretched hand. Okay. And which are the other bones that will fracture? Outstretched hand. Clavicle. So why it is occurring? Outstretched hand is going to give a flow. Weight টা দিয়ে হ্যান্ডেল করে তুলছে, ঠিক আছে, এবং হঠাৎ করে হচ্ছে সার্ভিস ফ্র্যাকচার। Next is the surgical neck of humerus. Okay. So which structure will damage? Now you know which structure will damage. Maxillary nerve. So paralysis of ethmoid and iris minor. Okay. And there will be involvement of posterior. The next one is okay, the next one is non-union. Non-union, what is the difference between non-union and non-union? কখন non-union দেখা যায়? Which age? After the age of 60. Okay. After the age of 60. So, which are the portion there will be non-union? Below the surgical neck. Okay, upper end of the shaft and the middle of the shaft. Okay, why? What is the portion of the nutrient artery? To find where the nutrient artery is? To find it out? Nutrient artery basics. Main thing is that what is the direction of nutrient artery? Nutrient artery কোথায় যাচ্ছে? উপরে যাচ্ছে, নিচে যাচ্ছে? জেনিটাল artery কি করছে? উপরে যাচ্ছে না নিচে যাচ্ছে? To the elbow I go, to the knee I feel. So if you are watering to the root of the tree, right, it will go to the upwards. Okay, so it will be always opposite. So in case of demand, মানে কি পানিয়ে যাওয়া, নিচটা কি উপরের দিকে উঠছে না নিচের দিকে নামছে? এখানে গ্রোইং হেড কোনটা হবে আপার হেড কিংবা এই টিবিয়ার ক্ষেত্রে কি হবে টিবিয়ার ক্ষেত্রে গ্রোইং হেডটা কোনটা নিচের দিকে কারণ ডিটা উপরে আছে সুতরাং নিচেটা উপর দিকে যাবে রাইট Then next one is the medial collateral ligament where it is attached medial collateral ligament of elbow. So it would be attached over here, right? So it is that much strong. If anyhow the elbow would be dislocated inferiorly, what will happen? There will be torn up of the medial epicondyle. Okay, the medial epicondyle would be separated. Understood? This medial collateral ligament it will be separating the medial epicondyle in case of inferior dislocation of. Okay. The medial collateral ligament is so much strong that in case of inferior dislocation of elbow, what is the word dislocation? So it is taking the medial epicondyle away from the bone. Okay. Okay. The next one is what is Hoffman ischemic contraction? So we in this form which one is the metaphysis? Metaphysis. Between the diaphysis and the metaphysis, right? So here which structure now should I told which structure is there on the lateral side? This is the medial side, this is the lateral side. Here is the situation of profound Okay. So in case of osteomyelitis, or supracondylar fracture, what will happen? There will be stagnant bacteria that will be lost over here, okay, causing the injury to the radial nerve and the brachial nerve. So what will happen? The forearm there will be rotten. Okay, so this is known as Hoffman ischemic contraction. They could not move the forearm. Okay, so this is all about the clinical anatomy of humerus. Another thing I told, shoulder joint dislocation would be inferiorly and anteriorly, but not superiorly. Why? Because of the tendon of biceps. ওপরে যাচ্ছে না শোল্ডার জয়েন্ট যখন ওকে কভার করে যাচ্ছে উপরে হচ্ছে ঠিক আছে এরিয়া কেন হচ্ছে না পোস্টেরিয়রলি শোল্ডার ডিসলোকেশনটা সব সময় ইনফিরিয়রলি হয় অ্যান্টিরিয়রলি হয় পোস্টেরিয়রলিও হয় না সুপিরিয়রলি হয় কেন So in case of shoulder dislocation it is occurring inferiorly and anteriorly. Okay, but it is not occurring superiorly or posteriorly. Why? Superiorly can dislocation হচ্ছে না superiorly posteriorly dislocation কেন হচ্ছে So এটা ইন্ডোর ক্যাভিটি রাইট তো superiorly কি আছে long head of bicep brachii সেটা superiorly হচ্ছে না posteriorly কেন হচ্ছে দেখ anteriorly মাত্র একটা মাসল অ্যাটাচ সাবস্ক্যাপুলারিস তিনটে মাসল পিছনে আছে তাহলে এটা ওকে so that is not about the disease okay so now two topic will cover okay one is what are the muscle of the forearm cubital fossa and muscle of the forearm Immediate process is easy, okay? Immediate process টা ডিসিশনে দেখাবো। So about the muscle of the front of thorax. So what are the muscles? At first it will be about the intramuscular septum, how it is attached. So এটা হচ্ছে flexor compartment হয়, extension compartment. So is it attached like this or something totally different is there? So it is attached completely. আমরা বলতে একটা flexion from medial border. ঠিক আছে? Medial border of forearm is we are flexing, right? And extension is occurring on the lateral surface, but not like this side or this side. Anterior posterior no. বোঝা যাচ্ছে? Medial and lateral. So in the flexor compartment, what are the muscles? আটটা muscles আছে, right? Superficial, intermediate. Superficial muscles are
flexor intermediate, radialis, flexor carbohydrate, retentalis, flexor.
So you have to tell on sequence. So okay, next we will discuss.